Hello student, today we will discuss about asexual reproduction. We have already seen different type of asexual reproduction: budding, binary fission, multiple fission, vegetative propagation, artificial and natural vegetative propagation, spore formation, etc. Now today we will see about the first one is parthenogenesis. If you split this word, you will get two words here, parthen plus genesis. Genesis means production and parthen means false. So it's a false production or false reproduction where offsprings are produced without fertilizing. You can say it's a modification of sexual reproduction where egg develops into a complete organism without any fertilization. So I can write the definition like this. It's production of complete organism without fertilization, because there is no mixing of DNA or no mixing of characteristic feature. That's why it's a uniparental reproduction and concluded as asexual mode of reproduction. Here egg cell, that is a haploid structure, that means and characteristic feature. If suppose this one is a human egg cell, then 23 chromosomes are found that undergoes in mitotic division and will form embryo. Although this embryo is haploid in nature again, and that give rise to haploid. This is called as parthenogenesis. It's very common in plants, but somewhere it's also found in animals. Natural parthenogenesis is found in honeybee. Three kinds of bees are found in a hive. They may be drone, worker, or third one is queen. Queen is a female that has ability to lay eggs. So it's a fertile female and it's 2N in nature. Worker is also a female, but it's a sterile female. It's also 2N in nature, while drone is a male, which is N in nature. So drones are produced by parthenogenesis, unfertilized egg of honeybee always give rise to drone. Just by using this method, we can also spray some hormones on carpel, where it will give rise to fruit but without seed. If we apply some growth hormone on carpel, that will develop into fruits, but because fertilization is not done, so seeds are not produced there. So this is a parthenogenesis known as parthenocarpy there. Carpy stands for carpel and partheno means false. So here the carpel give rise to fruit but without any seed form. Now the second category of asexual reproduction is known as micropropagation. It's artificial mode of asexual reproduction that can be done only in the laboratory. micropropagation is done in the laboratory condition where tissue culture is used to form different kind of plants with the help of some specific called as stem cells, because this one has ability to develop whole the plant. So we have to take totipotencies. Totipotency means complete potential to develop a plant or any organism. Cells of plant can be obtained by any kind of plant tissue, whether it's a leaf or stem or root. Isolated cells are called as explants. This experiment is always done in the sterilized condition. Some cells are isolated, this one is explant. Later on this explant is introduced in a culture media. This one is a culture media where nutritive material along with plant growth hormones are kept. This explant is introduced in this culture media. This process is called as inoculation. As soon as inoculation is over, this test tube should be covered up by a cotton plant. After the inoculation, allow this explant to grow there. After some time you will get cluster of cell. This is called as callus. Callus is also known as undifferentiated cell mass. Here the explant cell will obtain nutrition from the culture media and later on it grows and divides. After the division of cells, you will get a mass of cell that is undifferentiated, although work is not related yet, and have the capability to develop whole the plant. After the formation of callus, now differentiation gets started, and this differentiation gives rise to embryo-like structure which is known as embryoid. This embryoid is a heart-shaped structure. The upper part is responsible to form plumule while the lower part is responsible to form radicle. And after this, again it will obtain its nutrition continuously from the nutritive media. If there is a requirement, we can replace the nutritive media continuously, and later on plumule and radicles are formed. Now the small plant-like structure get arise, it's known as plantlet. It's a quite delicate structure. After hardening, the structure can be introduced in the soil to grow like a free plant. Hardening is a process to make it strong. In this condition, unfavorable conditions are introduced gradually and simultaneously. It will obtain the potential to develop in the unfavorable condition. This is called as hardening, and after hardening, this plantlet can be planted in any area, any soil also. This is micropropagation. That means development of complete plant with the help of a micro or a small segment. Thank you everyone.